My name is Matt Terrell from the University of Chicago and the Dreyfus Foundation Board. I'm really happy to have the opportunity to introduce Arup Chakraborty, who's the Robert T. Haslam Professor of Chemical Engineering, Chemistry, Physics, and Biological Engineering at MIT. He's also the director of the Institute for Medical Engineering and Science, which is a fairly new organization at MIT, and one of the founding members of the Reagan Institute, which is a joint operation of Massachusetts General Hospital, MIT, and Harvard that is really dedicated to accelerating the discovery of an HIV/AIDS vaccine, which is going to be the subject of Arup's talk here. Arup began his independent academic career at Berkeley in 1988, where he worked for 17 years before moving to MIT in 2005. He was, as it says in the program, a Camille Dreyfus Teacher Scholar in 1994. And Arup's career has really been exemplified by intellectual diversity. Early in his career, he worked on density functional theory of the electronic structure of polymers on surfaces toward adhesion problems. He moved, among other things, into work on zeolites and pattern recognition, which led him into some groundbreaking work on the theoretical description of immunological synapses. That's really the entree into the work, the theoretical work that he's been doing, giving insights on how to attack AIDS. I think he's a tremendous model of how to bring and merge powerful theoretical methods to important practical problems. Arup, the floor is yours.
Thank you, Matt. So I'm the barrier between you and cocktails now. So you know, the Camille Dreyfus Teacher Scholar Award was the most significant recognition I had received until 1994. So for that, I'm forever grateful. And therefore, I'm pleased to have this opportunity to describe some of our work today, all of which is focused on adaptive immunity. And by way of introduction, let me say that we have an adaptive immune system which is remarkable in that it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes, for which pathogen specificity should not be possible to pre-program, because it's not pathogen specificity to a fixed slate of known pathogens, but to unknown pathogens. So when you're infected by, say, a virus, it will hijack the transcriptional machinery of the host cell, thereby enabling synthesis of viral proteins and the assembly of many different virus particles which will go on to infect other cells. Now, important players in adaptive immunity are B cells. We have many clones of B cells, each with its own distinct receptor. And if the receptor on this particular B cell can bind sufficiently strongly to the spikes on this particular virus, then it can get activated, proliferate, make many copies of itself, and ultimately secrete soluble forms of this receptor, and that's what are called antibodies. These antibodies can then bind specifically to the spikes of this specific virus and dispose of it in a number of different ways, which I will not describe. But antibodies can only combat virus particles that are in your blood or extracellular spaces. They cannot attack infected cells or the virus particles that they harbor. Now, some of these proteins that are made of the virus, they are chopped up into short peptide fragments, about 10 amino acids long. And they are displayed on the surface of these infected cells, not by themselves, but in complex with another large human protein called MHC proteins. And that peptide MHC complex is the molecular flag of infection. Now, T cells are also important players in adaptive immunity. Again, we have many clones of T cells, each with its own distinct receptor called the T cell receptor, or TCR. And if the TCR on this particular T cell binds sufficiently strongly to this viral peptide, it can get activated, proliferate, and then mount an immune response in a number of different ways. For example, certain kinds of T cells, which are called cytotoxic T lymphocytes, or CTLs, are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it can then directly secrete products which will kill the infected cell and all the virus particles that they harbor. Now, after you've cleared an infection, then most of the B cells and T cells that proliferated in response to this particular infection actually die off. But some remain as so-called memory cells, which can mount robust and rapid responses upon reinfection with the same pathogen. And this immunological memory is the basis for all of vaccination. With a vaccine, what you try to do is induce antibodies and memory T cells that are specific for the pathogen against which you wish to protect the host. So as you might imagine, our interest in combating infectious disease and the untold amount of suffering caused by autoimmune diseases which emerge when adaptive immunity goes awry has led to a great deal of experimental research aimed toward understanding how adaptive immunity is regulated. And indeed, some spectacular discoveries have been made, especially over the last 40 years. But in spite of these major advances, an understanding of the principles that govern the emergence of an immune or autoimmune response have remained elusive. And just one practical consequence of this missing basic science is that over 30 years after the discovery of HIV, we still don't know how to make a vaccine against it. Now, in my view, a major barrier to this quest for mechanistic principles is that the pertinent processes involved in an immune response involve multiple different components that must act in highly cooperative collective ways in order for any phenomenon to emerge. Moreover, these processes span a spectrum of scales from the collective dynamics of many molecules within a single cell of the immune system to many types of cells cooperating in tissues and organs to the scale of the entire organism. It is this what I call hierarchically organized cooperativity with feedback between the scales that often makes it difficult to intuit underlying mechanism by looking at a few experimental reporters. And further confounding intuition is that many of these processes are inherently stochastic in character. So over the last 13 years, we've been trying to help confront this challenge by bringing together approaches that have traditionally been stovepiped in different disciplines. And in particular, what we've tried to do is bring together theory and computation that's rooted in physics and engineering with a diversity of different biological experiments and human clinical data. Now, the conceptual framework that we use most often in all our studies is statistical mechanics. As most of you know, the central goal of statistical mechanics is to understand how fluctuating, interacting microscopic objects can conspire together for coherent mesoscopic and macroscopic behavior to emerge. So even though statistical mechanics and immunology developed as parallel disciplines, i.e. no intersections, it is perhaps not surprising, given what I've told you about the challenges in immunology, that a convergence of these two disciplines has been fruitful. Now, these experiments that can test the predictions emerging from the theoretical and computational work are all carried out in the laboratories of our collaborators, who are all immunologists at various medical schools. With the key assistance of these collaborators, indeed teachers, I would say, we've had some luck over the years in shedding light on some basic questions in immunology and more recently on human disease. So we've been interested for a long time on how the engagement of the T cell receptor to viral peptides gets translated to functional activation by the collective dynamics of these intracellular proteins. We've also been interested in how development of T cells in an organ called the thymus leads to this amazing ability of specificity for unknown pathogens. But the work I'll describe to you today concerns a scourge on this planet. Over the last 4 years, I've made frequent visits to Africa, and that has made vivid to me the havoc that HIV/AIDS is wreaking in that part of the world. Just as one example of a statistic, Here are data from collaborators in a hospital in Durban, South Africa, where they test pregnant women for HIV. And as you can see, in this KwaZulu-Natal province of South Africa, by the time you're a 24-year-old woman, you have a 65% chance of being HIV positive. Statistics like this, combined with the poverty that you see there, make clear that a vaccine is urgently needed. Antiretroviral therapy is not, in these parts of the world, a viable solution today. Now, a major barrier to this quest for a HIV vaccine has been the high mutability of the virus. So in these phylogenetic trees, the number of N's that you see corresponds to the diversity of the virus strains that are circulating. And as you can see, the diversity of the flu virus in the entire world in a particular year is comparable to the diversity of HIV strains in a single infected patient, and completely dwarfed by the diversity of strains in just one part of Africa. Now, this high mutability poses a challenge for either an antibody or a T-cell arm of a vaccine. A vaccine-induced antibody is supposed to bind specifically to the HIV viral spike. But if a mutation emerges in the protein that makes up the spike, it will abrogate this recognition, making this vaccine-induced antibody not very useful. Similarly, for a T-cell arm of a vaccine, memory T cells are supposed to bind specifically to HIV peptides. But if a mutation emerges in the protein from which this peptide is derived, it will abrogate that binding, making this vaccine-induced memory T cell not very useful. Now, recent discoveries, especially in the last 3 or 4 years, suggest that these challenges can be confronted and perhaps even overcome. In the interest of time, I won't take you through some of the highlights of these discoveries, but rather I will focus on some of our own work which pertains to a possible T-cell arm of a therapeutic or prophylactic vaccine, or as a metaphor, how to hit HIV where it hurts with T cells. So the first part of the work I'll describe has already been published, and it was led by 2 people in my group, Vincent Dahirel, who was a postdoc at the time, but now has his own faculty position at the Curie Institute in his native Paris, and Karthik Shekhar, who is still a graduate student at MIT. And it's a very close collaboration with Bruce Walker's lab. Bruce is a clinician scientist at the Mass General Hospital. Now, because of the high mutability of the virus, there has been interest in focusing a vaccine-induced immune response to target single amino acids in HIV proteins that appear highly conserved when you look at protein sequences of the same protein derived from virus samples extracted from many patients. The idea being that if you hit with immune response here, the virus has to make a mutation there to evade the immune response. But if it makes a mutation there, it's likely to be bad for the virus, because otherwise, why else do you see it so conserved when you look at these protein sequences? But this strategy has been of very limited efficacy, at least when you look at humans or monkeys that naturally target such residues. Because while all the things I just told you do happen, very quickly HIV evolves other mutations, so-called compensatory mutations, that restore the fitness of the virus. So now you have escaped the immune pressure by the primary mutation, and then you made other mutations that have made you just as fit as before. Now, if you think about it, this way of thinking about conservation is really a one-dimensional way of thinking about conservation. Because what you're doing is you're looking at a single residue and asking, how do mutations in this single residue influence viral fitness? Then you look at another one independently, and then you ask, which site or sites are ones where mutations are more likely to be deleterious for the virus? But this way of thinking ignores coupling between the mutations, such as these compensatory mutations. So we thought it might be more useful to see if we could take HIV polyproteins and divide them up into groups of amino acids, a lower dimensional representation, if you like, which is such that amino acids within a group co-evolve, in that combinations of mutations in amino acids within a group collectively influence viral fitness, but amino acids in different groups evolve essentially independently of each other. If this were possible, then one could hope that in some group or groups compared to other groups, a greater proportion of the combinations of mutations are bad for the virus. Such a group would be immunologically vulnerable to multiple points of immune attack, because multiple points of immune attack would promote the emergence of multiple mutations to escape the T cell pressure, but that was likely to make the virus unfit because multiple mutations in this group are more likely to be bad. Notice how this effect is unlikely to be compensated by mutations in amino acids in another group, because amino acids in different groups evolve essentially independently of each other. But do such groups exist in HIV proteins? Well, in principle, you should be able to answer this question by analyzing the various sequences of HIV proteins that I told you are available. But the problem is complicated by two effects. One is there is noise. After all, you still have a finite sample of sequences, so there's statistical noise in the correlations. The other is that some collective correlations that you see are not because of reasons of any intrinsic viral fitness, but simply reflect the fact that they're all HIV proteins or phylogeny. So recently, we think we've had some luck in deconvoluting these confounding effects and identify these groups of amino acids that are collectively coupled because of reasons of intrinsic viral fitness. And in the first part of the work I'll describe, we used a method called random matrix theory, which was first invented by Eugene Wigner, a chemical engineer turned theoretical physicist, I might add, who developed this in the context of high energy physics. But random matrix theory has been used in many realms of physics to analyze stock market price fluctuations, and also more recently by Stan Leibler to think about a particular enzyme. Let me tell you what Gene Stanley and others did when they used random matrix theory to analyze stock market price fluctuations. At the end of the day, they were able to take all the companies listed in the stock market and divide the companies up into groups of companies. Price fluctuations of companies within a group were collectively coordinated, but the prices of companies in different groups fluctuated independently. Now, when we looked at the identities of the companies within each of these groups, they found that they had rediscovered the well-known sectors of the economic markets, because one of the groups was made up of all the auto companies and auto parts companies, another was all the hardware and software companies, another were all the financial sectors, and so forth. So we reasoned that since our intuition about HIV sectors, if you like, is not so well developed, we might learn something useful along the lines of what I told you by doing some analogous analysis. So the data we have are sequences of the same HIV polyprotein extracted from different viruses from different patients. So you could go to a particular residue, let's call it J, and ask if in this particular sequence there is a mutation away from the most probable amino acid at residue J, is that correlated with a similar mutation arising in residue Y? Then you can average this information over all sequences to obtain an estimate of the correlation between mutations at this pair of sites. By doing this for each pair, you go from lining up all the sequences to a pair correlation matrix. Now, each element of this matrix is really measuring the difference in the frequency with which you see the double mutation minus the frequency with which you would see the double mutation if these mutations were occurring independently. So it's really measuring the coupling between the mutations. Now, the orthogonal eigenvectors of this matrix contain information on collective correlation that go way beyond pairs, doubles, triples, higher order things. But this information is corrupted by noise and phylogeny. And so that's what we cleaned out in order to obtain the real couplings on higher orders. So what do we actually do? So what you do is you just diagonalize this matrix and find all its eigenvalues. And so I'm going to focus today's talk only on one HIV polyprotein called GAG in the interest of time. And this is the eigenvalue spectrum you obtain by doing this analysis for GAG. So I call it a spectrum because this is the number of eigenvalues with a particular value of the eigenvalue. Now, Mr. Wigner's theorems from random matrix theory tell us that for the number of sequences we have and the number of amino acid positions in GAG, which is 500, if this matrix was simply reflecting random noise, the largest eigenvalue should be at 2.4. But this theorem is in the limit where the number of sequences and the number of amino acid positions is infinitely large, which we don't have. So to assess finite size effects, what you can do is you go to the sequence alignment and go to any particular site and randomly permute all the amino acids and do that for every amino acid position. Now you've generated a randomized sequence. Find the eigenvalues again. Do it again and again, and you find that the eigenvalue spectrum for such randomized sequences of GAG looks like that. So Mr. Wigner's theorem is pretty accurate, right? So you've got a couple of eigenvalues more than 2.4, but none above 2.7. So all the eigenvectors below this limit are corrupted by noise, and we don't interpret them. Now, whenever we analyze HIV polyproteins by this method, we always find that there's one eigenvalue that's much larger than all the others, and it's also a coherent mode. And I have an analytical theory which says that if phylogeny was the only game in town other than noise, then there would be only one eigenvalue above the noise limit, and it would scale with the length of the protein, which is 500 in this case, and therefore it would be very large, and it would be a coherent mode. So we think that this particular eigenvector is very badly corrupted by phylogeny. Then by parsing the eigenvectors associated with the next few eigenvalues, we obtain these collectively co-evolving groups. And in the case of GAG, we find 5 such collectively co-evolving groups, and I shall call them sometimes sectors in analogy with the economic markets. Now, the first thing you could do is you could ask, is it possible that one of these 5 groups, the amino acids contained in it, just happen to be much more conserved compared to other residues at the single residue level? That is, the single point level, this group has much more conservation. You can answer this question because I know the amino acids in each of these groups. And this is what you find. This is the average frequency of conservation at the 1 point level for amino acids in group 1, 2, and so forth. And as you can see, the sectors are not distinguished by this metric. This one's slightly lower, but this guy has only 14 amino acids in it. But if you now look at the signs of the correlations that describe the character of multiple simultaneous mutations occurring in amino acids within a group, differences between the sectors emerge. What do the signs mean? So if a multi-point correlation function is positive, it means that the multiple mutation arises more frequently than if the mutations were occurring independently, suggesting that the multiple mutant is more fit than it should be by just independent mutations. So these could correspond to these compensatory mutations I was describing. So you don't want to hit regions or sectors that are have a lot of positive correlations with immune pressure, because there are many escape pathways. A negative correlation between a multiple mutation means that the multiple mutant arises far less frequently than if the mutations were arising independently, suggesting that the multiple mutation is far less fit than you would expect from independent mutations. So regions rich in negative correlations is where you want to hit with immune pressure at multiple points, because there's not many paths out. So by this metric, you find that one particular sector, sector 3, has the highest proportion of negative to positive correlations. And by this metric, sector 1 is second. Now, I know which amino acids are in sector 3. There are 57 of them. 52 of them are in a single protein that is excised out of GAG. GAG's a big polyprotein. It gets made as a big polyprotein and then it's chopped up into shorter bits. And one of the shorter bits is P24, which has 231 amino acids. And 52 of these 57 sector 3 residues are in this one protein. This is the structure of that protein. And the blue balls are those 52 sites that make up sector 3. So when we first saw this, we were very disappointed, because there's no way we could tell why this clump of amino acids and this clump were co-evolving. But 6 of these P24 proteins make hexamers. The hexamers make interfaces with other hexamers. And that's what tiles the viral capsid that contains the RNA genome of HIV. The structure of this hexamer is known. I show it to you here. 1 P24, second, third, fourth, fifth, and sixth. And what Vincent has done here is he has taken those 52 residues and has partitioned them into 3 classes: red, green, and purple. The reds and greens are more than 70% of those 52 sites. So they're the preponderance of sites. Even without doing analysis, you can see, which we have done, that the red residues are forming some, but not all, critical interfaces between P24 proteins in forming the hexamer. Similarly, the structure of the hexamer-hexamer interface shows that the green residues are forming hexamer-hexamer interfaces. So notice how this analysis has picked out collective correlations that emerge not from the structure of a single protein, but from the importance of assembling multi-protein structures that are critical for viral function. In this case, capsid assembly. Notice also that now you can see why there are so many more negative correlations in these regions. While 1 or 2 mutations in these interfaces might still keep the interfaces stable, multiple mutations are likely to destabilize these interfaces. Now remember I told you about this sector 1. Those guys end up largely composing the core of this hexamer. Now, I won't take you through the details of the mathematical analysis, but all our analysis suggests that this sector 3, i.e. this interface guys, is much more what I call multidimensionally conserved than sector 1. That is, they are much more vulnerable to multiple mutations than this sector 1, which forms sort of the core of the hexamer. And this will become important in a few minutes. Okay, so this is interesting to us anyway, but totally worthless for vaccinology if human MHC molecules cannot display peptides from these vulnerable regions, because as I told you early on, what the T cell sees through its T cell receptor are viral peptides bound to these MHC molecules. Now we each have about 6 to 12 types of these MHC proteins. And the MHC gene is the most polymorphic gene in humans. That is to say, there are tens of thousands of variants in the population. So the chance that 2 individuals have exactly the same set of MHC molecules is about 0. There's certainly none in this room, a pair in this room, because I don't see any identical twins. Now, there are a group of people who are called elite controllers. They are able to control HIV infections naturally with their immune system. And all evidence suggests that they do so with their T cell response. Now patients, groups of patients who are elite controllers are statistically enriched with people who possess certain MHC molecules whose names are listed there. So if you have the B57, for example, you're very likely to be an elite controller. Now, any given MHC molecule will display many different HIV peptides, but a few are called immunodominant because those few viral peptides for this MHC attract most of the T cell attention. So the immunodominant peptides for these MHC molecules associated with elite control are known. So we went and asked the following question. Where are the amino acids in the peptides that are in the immunodominant peptides of these elite controller MHCs vis-a-vis our sector picture. So remarkably, the sector with the greatest proportion of sites targeted by people who are elite controllers is sector 3. Moreover, people with these genes do target multiple points. For example, if you have the B57 MHC molecule, you are most likely to be an elite controller. People with this MHC are targeting 8 out of those 52 residues. Now last year, a couple of years ago, by bringing together some methods from statistical physics with human clinical data, we published a paper in one of the tabloids where we suggested that people who have this B57 molecule, their T cells are more likely to recognize point mutations of peptides that they do target. So they're more cross-reactive. Notice how this cross-reactivity further augments this multiplicity of attack. This is true for the other genes too. I won't tell you about them today. But let me just remark first that this picture is a little bit misleading because it says sector 4 and 5 are targeted 10% of the time. Well, sector 4 has 10 residues, so it's targeted at 1 point when it's targeted at 10%. And sector 5 has 14, so again 1 point. So these guys are not targeted at multiple points. But sector 1 has 80 residues, so it is targeted at multiple points by these elite controllers. So this suggested another way to test our ideas. If 2 regions of the proteome are equally targeted by the immune response, all other things being equal, you would imagine that the number and types of mutations in both these regions would be similar because they are attacked and there's no difference between them. But I told you earlier that sector 3 is much more multidimensionally conserved than sector 1. So our prediction was that if you went to elite controller patients and you took virus samples out of them and you sequenced them, you would find that even though sector 3 is targeted more than sector 1, there should be far fewer multiple mutations in sector 3 compared to sector 1. Notice also the prediction that as far as single point mutations were concerned, sector 1 and 3 should be the same, because I showed you earlier on at the single point level these groups of amino acids were no different. So Bruce did this. He went to 103 elite controller patients that he has. He took virus samples out of them and he sequenced them. And this is the data. This is the number of unique strains of HIV in these patients with 1 mutation, 2, 3, 4, etc., parsed according to sector 1 in blue and sector 3 in red. As you can see, at the 1 point level, they're roughly the same. Double mutations, far fewer in sector 3. Triple, far fewer. 4s and 5s, none. So we think taken together, what I've said so far provides strong support for our idea that if you can identify these collectively coordinated groups of amino acids where multiple mutations are much more likely to be bad for the virus, and you hit it with multiple points of immune pressure, you will corner the virus between a rock, the immune pressure, and a hard place, which is unfit virus. But what about people with other MHCs, most of us? Well, the fact is that diverse people with diverse MHCs present peptides from these vulnerable regions, not just the GAG part I've shown you, but also others. But they do so subdominantly. That is, they present them, but when other bits are present, T cells mostly attack the other bits. So this suggests to us that the vaccine against a bug like HIV, the active component of the immunogen should not be the usual thing, which is whole proteins, because they distract the immune system. But rather, the immunogen should be made up of bits that satisfy 2 criteria simultaneously. 1, it maximizes sites in these vulnerable regions, and it maximizes sites in subdominant and dominantly presented peptides presented by MHCs that span a wide swath of a target population, like African population or Caucasian population. Because if that's the immunogen, then the memory T cell response that you will generate will be only for these vulnerable regions. Why? Because the other parts ain't there. Now, when you get a natural infection, of course, you get the whole thing. But as I told you early on, the memory response goes first, and so early on you will attack the virus only in the vulnerable regions before this ineffective response gets going. And we know that both in vaccine context and otherwise what happens early on determines outcome. So we have been busy designing these immunogens, and we ran into a little problem. Even in these vulnerable regions, there are escape pathways still there. There are just fewer of them. So how do we block them? So that's the problem that former postdoc Andy Ferguson worked on. He's now a professor at Illinois. And what Andy wanted to do was very ambitious. He wanted to translate the sequence data to quantitative viral fitness landscapes. What's a viral fitness landscape? So just for illustration, imagine the protein made up of 2 amino acids. Otherwise, I can't draw it in a 2-dimensional picture. So that's one amino acid, another one. And each point here is a different mutation. Each point here is a different mutation. So any point in that 2-dimensional plane is a different viral strain. And so Andy wanted to say, give me a strain, I'll tell you its fitness. Having this fitness landscape can really inform rational immunogen design, because you want to push the virus off from this hill into a valley, but you also want to block the mountain passes that HIV has to traverse by mutation to go from one hill to another when this hill is under attack. So I think Andy has succeeded in developing these fitness landscapes. Again, I won't take you through the details, but following work by Bill Bialek in the context of neurobiology, you can show that the least biased or maximum entropy model that fits the 1 and 2 point mutational probabilities that you see in the sequence data is described by the Ising spin glass Hamiltonian. This is the only thing in my talk today that I was still thinking about in 1994 when I got the Dreyfus Scholar Award. Anyway, so you can then fit the coupling constants Hi and Jij to the data, and then you've got all of spin glass theory to describe these fitness landscapes. But is it right? I mean, do you really get the right fitness landscapes? Well, so we've tested it against experiment. So the metric of fitness that we get is a quantity called E. You give me a sequence, and he'll give you a value of E. And the theory says that E minus the energy corresponding to the wild-type viral sequence should correlate negatively with the log of the fitness of the mutant divided by the fitness of the wild-type. So we made a number of predictions, and our collaborators in Africa made those mutations in the viruses and measured their replicative fitness in vitro. So this is really intrinsic viral fitness, not in the presence of other people's immune pressure. So here's the comparison of the data. And you see, except for these 2 outliers, this is pretty good. And this morning I got 11 more data points. 10 of them fit on this as well. Now this is all data from one lab. Without detail, in the interest of time, let me just say that we've also looked at all the available data. And again, you can see the statistical accuracy with which we predict these fitness. Well, this is a postdiction, and this is a prediction. The 2 are not synonyms. So. And if you take out these 2 measurements from the same lab, things get even better. We've also tested this against real human clinical data, which I will not take you through, except to mention just one here. So there is data now on 6 patients at Mass General Hospital where they follow the evolution of the virus in single patients over time. So it's only 6 patients, okay? But for these 6 patients, given the immune pressure they are putting, what we predict to be the highest fitness paths on this fitness landscape is what they follow. So at some level, we are very happy that we can predict these fitness landscapes. But it also brought us to a question. Why are we being so good at predicting these fitness landscapes? After all, the sequences from which we inferred the fitness landscape, are not an equilibrium ensemble of sequences that is sampling this spin glass Hamiltonian. In fact, each sequence is the product of a highly non-equilibrium host pathogen dynamics in each individual. So why are we getting the intrinsic fitness landscapes? Different people, we seem to be correct. External in vitro measurements, we seem to be correct. So I'll close by simply mentioning that with my physics colleague Mehran Kardar and the ubiquitous Karthik Shaker, we have thought about this question. And I won't take you through the details except to note that by using a mapping of Eigen's evolution of virus quasi-species to the statistical mechanics of a two-dimensional Ising model, we've been able to show, first, that we do not get the fitness landscape correctly compared to if we really did have the equilibrium ensemble. But we get the right order of fitnesses. So if you take all the sequences, what you would get from equilibrium ensemble would be the same order of fitnesses. So we get the fitness landscape within a constant. That's why when we compare with the experiment, it comes out right, because there's this constant in front that it just defines where the intercept of that thing is. So we're a little bit excited about this, because now we may have a way of getting fitness landscapes of viruses to do rational immunogen design for diverse viruses, not just HIV. So now, anyway, let me finish by saying that both our sector ideas and these fitness landscapes, we have designed antigens which we think should be a potent immunogen for HIV. There's nothing to look at in this slide other than they exist. So what's happening now is that these whole proteins give you a big inflammatory response, whereas these long peptides have a hard time giving you an immunogenic response. So you have to optimize the delivery method. So this is all out of my hands now. But these guys over here are now optimizing the adjuvant and delivery method in mice whose immune systems are supposedly mimic that of humans, not normal mice. And then once that is over, that will be tried in both non-human primates and humans. But in fact, I discovered recently that a Swiss group has actually put this, a more primitive version of our published antigen, into humans who are already infected, and it has brought down their viral load just a bit. But they didn't deliver it that in an optimal fashion, but still. So we are hopeful that this may be useful. On the other hand, it might be a complete dud. But nonetheless, I will close with that, and I think I've thanked everybody who contributed to this work. Thank you.
Coming close to the end of our time, but I think we could entertain one or two questions if there are some. Arup will be here, so you can talk to him afterwards. Please.
All right, thanks. Very inspiring talk. So when you were talking about the different sectors and how your modeling actually predicted that, I was just wondering that these sectors have got a time sensitivity. In other words, if you take data from different times, then you wouldn't have that correlation. Whereas the patients that were taking, their infection times might have been different. So the question is, if a sample that contained patients with similar infection types were used, would your prediction even get better?
No, I think I understand your question perfectly well, but I think this problem is dealt with because all of these sequences were collected from people in the so-called chronic stage of infection. So in the chronic stage of infection is that portion after some time when the viral load has gone up, comes down again, and has stayed steady for various periods of time. So in that period, all the samples are from people at that stage of infection.
Go ahead, Greg. You'll be the last one.
This is absolutely beautiful and inspiring, Arup. It's clear that it works. And
not yet.
Well, I mean, at least it looks like it works mathematically. But I'm curious.
No, it works in humans too, but not in the vaccine context.
Okay, I see that it works, but I'm curious about the generality of the approach to other viruses, that the eigenvalues of a two-body correlation matrix projects only on a subset of the n-body space. And it seems that, I mean, we can couch this in the language of escape routes, but have you looked at the n-body correlations specific to the subspace targeted by your sector to see if there's a way to select which pieces of these 52, for example, to target most profitably?
Yeah, that's an excellent question, and that's how we designed the antigens that I showed you, but not with the sector approach. we did it actually with the fitness landscapes that we extracted, because now you can manipulate that much better. So what you do now is you just take projections on different planes defined by where you can put immune pressure and where the multiple mutations have the largest negative value of those JIJ couplings.
Thank you.
Okay, thanks.